Counterillumination camouflage. Principle of counterillumination camouflage in firefly squid, Wattisenia scintillans. When seen from below by a predator, the bioluminescence helps to match the squid's brightness and color to the sea surface above. In many animals of the deep sea, including several squid species, bacterial bioluminescence is used for camouflage by counterillumination, in which the animal matches the overhead environmental light as seen from below. 49. In these animals, photoreceptors control the illumination to match the brightness of the background. 49. These light organs are usually separate from the tissue containing the bioluminescent bacteria. However, in one species, Euprymnoscolopes, the bacteria are an integral component of the animal's light organ. 50. Attraction. Storoteuthis cirtensis bioluminescent photophores. Bioluminescence is used in a variety of ways and for different purposes. The serrate octopod Storoteuthis cirtensis emits bioluminescence from its sucker-like structures. 51. These structures are believed to have evolved from what are more commonly known as octopus suckers. They do not have the same function as the normal suckers because they no longer have any handling or grappling ability due to its evolution of photophores. The placement of the photophores are within the animal's oral reach, which leads researchers to suggest that it uses its bioluminescence to capture and lure prey. 52. Fireflies use light to attract mates. Two systems are involved according to species. In one, females emit light from their abdomens to attract males. In the other, flying males emit signals to which the sometimes sedentary females respond. 48-53. Click beetles emit an orange light from the abdomen when flying and a green light from the thorax when they are disturbed or moving about on the ground. The former is probably a sexual attractant but the latter may be defensive. 48. Larvae of the click beetle Pyrophorus nyctiphanus live in the surface layers of termite mounds in Brazil. They light up the mounds by emitting a bright greenish glow which attracts the flying insects on which they feed. 48. In the marine environment, use of luminescence for mate attraction is chiefly known among ostracods, small shrimp-like crustaceans, especially in the family Cyprididae. Pheromones may be used for long-distance communication, with bioluminescence used at close range to enable mates to home in. 33. A polychaete worm. The Bermuda fireworm creates a brief display, a few nights after the full moon, when the female lights up to attract males. 54. Defense. Acanthophyra purpurea has photophores along its body which it uses in defense against predators. The defense mechanisms for bioluminescent organisms can come in multiple forms: startling prey, counter-illumination, smoke screen or misdirection, distractive body parts, burglar alarm, sacrificial tag or warning coloration. The shrimp family Oplophoridae use their bioluminescence as a way of startling the predator that is after them. 55. Acanthophyra purpurea, within the Oplophoridae family, uses its photophores to emit light, and can secrete a bioluminescent substance when in the presence of a predator. This secretory mechanism is common among prey fish. 55. Many cephalopods, including at least 70 genera of squid, are bioluminescent. 33. Some squid and small crustaceans use bioluminescent chemical mixtures or bacterial slurries in the same way as many squid use ink. A cloud of luminescent material is expelled, distracting or repelling a potential predator, while the animal escapes to safety. 33. The deep-sea squid Octopoteuthis delatron may autotomize portions of its arms which are luminous and continue to twitch and flash, thus distracting a predator while the animal flees. 33. Dinoflagellates may use bioluminescence for defense against predators. They shine when they detect a predator, possibly making the predator itself more vulnerable by attracting the attention of predators from higher trophic levels. 33. Grazing copepods release any phytoplankton cells that flash. Unharmed, if they were eaten they would make the copepods glow, attracting predators, so the phytoplankton's bioluminescence is defensive. The problem of shining stomach contents is solved, and the explanation corroborated. In predatory deep-sea fishes, their stomachs have a black lining able to keep the light from any bioluminescent fish prey which they have swallowed from attracting larger predators. 9. The sea firefly is a small crustacean living in sediment. At rest it emits a dull glow but when disturbed it darts away leaving a cloud of shimmering blue light to confuse the predator. During World War II it was gathered and dried for use by the Japanese army as a source of light during clandestine operations. 16. The larvae of railroad worms, Phrixothrix, have paired photic organs on each body segment, able to glow with green light. These are thought to have a defensive purpose. 56. They also have organs on the head which produce red light. They are the only terrestrial organisms to emit light of this color. 57. Warning. Aposematism is a widely used function of bioluminescence, providing a warning that the creature concerned is unpalatable. It is suggested that many firefly larvae glow to repel predators. Some millipedes glow for the same purpose. 58. Some marine organisms are believed to emit light for a similar reason. These include scale worms, jellyfish and brittle stars, but further research is needed to fully establish the function of the luminescence. Such a mechanism would be of particular advantage to soft-bodied cnidarians if they were able to deter predation in this way. 33. The limpet Latia neretoides is the only known freshwater gastropod that emits light. It produces greenish luminescent mucus which may have an anti-predator function. 59. The marine snail Hinia brasiliana uses flashes of light, probably to deter predators. The blue-green light is emitted through the translucent shell, which functions as an efficient diffuser of light. 60. Communication. Pyrosoma, a colonial tunicate, each individual zooid in the colony flashes a blue-green light. Communication in the form of quorum sensing plays a role in the regulation of luminescence in many species of bacteria. Small extracellularly secreted molecules stimulate the bacteria to turn on genes for light production when cell density, measured by concentration of the secreted molecules, is high. 33. Pyrosomes are colonial tunicates, and each zooid has a pair of luminescent organs on either side of the inlet siphon. When stimulated by light, these turn on and off, causing rhythmic flashing. No neural pathway runs between the zooids, but each responds to the light produced by other individuals, and even to light from other nearby colonies. 61. Communication by light emission between the zooids enables coordination of colony effort, for example in swimming where each zooid provides part of the propulsive force. 62. Some bioluminescent bacteria infect nematodes that parasitize Lepidoptera larvae. When these caterpillars die, their luminosity may attract predators to the dead insect, thus assisting in the dispersal of both bacteria and nematodes. 48. A similar reason may account for the many species of fungi that emit light. Species in the genera Armillaria, Mycena, Omphalodus, Pinellus, Pleurotus and others do this, emitting usually greenish light from the mycelium, cap and gills. This may attract night-flying insects and aid in spore dispersal, but other functions may also be involved. 48. Quantula striata is the only known bioluminescent terrestrial mollusk. Pulses of light are emitted from a gland near the front of the foot and may have a communicative function, although the adaptive significance is not fully understood. 63. Mimicry. A deep-sea anglerfish, Buphoceratias wedley, showing the esca lure. Bioluminescence is used by a variety of animals to mimic other species. Many species of deep-sea fish such as the anglerfish and dragonfish make use of aggressive mimicry to attract prey. They have an appendage on their heads called an esca that contains bioluminescent bacteria able to produce a long-lasting glow which the fish can control. The glowing esca is dangled or waved about to lure small animals to within striking distance of the fish. 33. 64. The cookie-cutter shark uses bioluminescence to camouflage its underside by counter-illumination, but a small patch near its pectoral fins remains dark, appearing as a small fish to large predatory fish like tuna and mackerel swimming beneath it. When such fish approach the lure, they are bitten by the shark. 65. 66. Female Photorus fireflies sometimes mimic the light pattern of another firefly, Photinus, to attract its males as prey. In this way they obtain both food and the defensive chemicals named lucibuphagins, which Photorus cannot synthesize. 67. South American giant cockroaches of the genus Lucihormetica were believed to be the first known example of defensive mimicry, emitting light in imitation of bioluminescent, poisonous click beetles. 68. However, doubt has been cast on this assertion, and there is no conclusive evidence that the cockroaches are bioluminescent. 69. 70. Flashing of photophores of black dragonfish, Mollicosteus niger, showing red fluorescence. Illumination. While most marine bioluminescence is green to blue, some deep-sea barbelled dragonfishes in the genera Aristostomias, Pachystomias and Mollicosteus emit a red glow. This adaptation allows the fish to see red pigmented prey, which are normally invisible to other organisms in the deep ocean environment where red light has been filtered out by the water column. 71. These fish are able to utilize the longer wavelength to act as a spotlight for its prey that only they can see. 71. The fish may also use this light to communicate with each other to find potential mates. 72. The ability of the fish to see this light is explained by the presence of specialized rhodopsin pigment. 71. The mechanism of light creation is through a suborbital photophore that utilizes gland cells which produce exergonic chemical reactions that produce light with a longer, red wavelength. 73. The dragonfish species which produce the red light also produce blue light in photophore on the dorsal area. 73. The main function of this is to alert the fish to the presence of its prey. 74. The additional pigment is thought to be assimilated from chlorophyll derivatives found in the copepods which form part of its diet. 74. The angler siphonophore Arena utilizes red bioluminescence in appendages to lure fish. 73. Biotechnology. Biology and medicine. Bioluminescent organisms are a target for many areas of research. Luciferase systems are widely used in genetic engineering as reporter genes, each producing a different color by fluorescence, 75, 76, and for biomedical research using bioluminescence imaging. 77, 78, 79. For example, the firefly luciferase gene was used as early as 1986 for research using transgenic tobacco plants. 80. Vibrio bacteria symbiose with marine invertebrates such as the Hawaiian bobtail squid, Euprymna scolopes, are key experimental models for bioluminescence. 81, 82. Bioluminescent activated destruction is an experimental cancer treatment. 83. In vivo luminescence cell and animal imaging uses dyes and fluorescent proteins as chromophores. The characteristics of each chromophore determine which cell areas will be targeted and illuminated. 84. Light production. A firefly, Petunia, genetically engineered to produce luciferase. The structures of photophores, the light-producing organs in bioluminescent organisms, are being investigated by industrial designers. Engineered bioluminescence could perhaps one day be used to reduce the need for street lighting, or for decorative purposes if it becomes possible to produce light that is both bright enough and can be sustained for long periods at a workable price. 11. 85, 86. The gene that makes the tails of fireflies glow has been added to mustard plants. The plants glow faintly for an hour when touched, but a sensitive camera is needed to see the glow. 87. University of Wisconsin-Madison is researching the use of genetically engineered bioluminescent E. coli bacteria, for use as bioluminescent bacteria in a light bulb. 88. In 2011, Philips launched a microbial system for ambience lighting in the home. 89, 90. An IGAM team from Cambridge, England, has started to address the problem that luciferin is consumed in the light-producing reaction by developing a genetic biotechnology part that codes for a luciferin regenerating enzyme from the North American firefly. 91. In 2016, Glowy, a French company, started selling bioluminescent lights for shop fronts and street signs. 92. For use between 1 and 7 in the morning when the law forbids use of electricity for this purpose. 93. 94. They use the bioluminescent bacterium Alivibrio fischeri, but the maximum lifetime of their product was 3 days. 92. In April 2020, plants were genetically engineered to glow more brightly using genes from the bioluminescent mushroom Neonathopanus nambi to convert caffeic acid into luciferin. 94. 95. Another possible application is to replace chemiluminescence with bioluminescent enzymes. A Canadian company, Lux Bio, is developing long-duration bioluminescent enzymes for this purpose. 96. 97. ATP bioluminescence. ATP bioluminescence is the process in which ATP is used to generate luminescence in an organism, in conjunction with other compounds such as luciferin. It proves to be a very good biosensor to test for the presence of living microbes in water. 98. 99. Different types of microbial populations are determined through different sets of ATP assays using other substrates and reagents. Ranilland-Gaussier-based cell viability assays use the substrate cholinterazine. 100. See also. Animal coloration. Biophoton. Life That Glows, 2016 full-length documentary. Notes. A. However, the name phosphorus, as used in the 17th century, did not necessarily mean the modern element. Any substance that glowed by itself could be given this name, meaning light bearer. 14. References. 1. Pless, Merrick, the 11th of November 2021. Lab snapshots by Merrick Pless. Microbiology: The biology on a different level. WeirdScience.eu. Retrieved the 2nd of July 2023. 2. Callaway, E. 2013. Glowing plants spark debate. Nature, 498, 15-16, the 4th of June 2013. 3. Smiles, Samuel, 1862. Lives of the Engineers. Volume 3. George and Robert Stevenson. London, John Murray. p. 107. ISBN 978-0-7153-42817. ISBN refers to the David and Charles reprint of 1968 with an introduction by L.T.C. Rolt. 4. Fries, Barbara, 2006. Cole, A Human History, RCKC and PG equals PA 51. Arrow, p. 51. ISBN 978-0-09-947884-3. 5. Harvey cites this as Baker, J. 1743-1753. The Microscope Made Easy and Employment for the Microscope. Bioluminescence. Lepidoptera migration. Many populations of Lepidoptera, butterflies or moths, migrate, sometimes long distances, to and from areas which are only suitable for part of the year. Lepidopterans migrate on all continents except Antarctica, including from or within subtropical and tropical areas. By migrating, these species can avoid unfavorable circumstances, including weather, food shortage, or overpopulation. In some Lepidopteran species, all individuals migrate, in others, only some migrate. Monarch butterfly tagged to track its migration. Danaus plexippus migration map in North America. Tyriomala septentrionis migrate in millions between Eastern Ghats and Western Ghats in India. Catopsilia pomona migrate regularly in South India and Himalayas. The best-known Lepidopteran migration is that of the eastern population of the monarch butterfly, which migrates from southern Canada to wintering sites in central Mexico. In late winter, early spring, the adult monarchs leave the transvolcanic mountain range in Mexico for a more northern climate. Mating occurs and the females begin seeking out milkweed to lay their eggs, usually first in northern Mexico and southern Texas. The caterpillars hatch and develop into adults that move north, where more offspring can go as far as central Canada until next migratory cycle. The Danaids in South India are prominent migrants, between the Eastern Ghats and Western Ghats. 3 species will be involved in this, namely Tyriomala septentrionis, Euploea cor, and Euploea sylvester. Sometimes they are joined by lemon pansy, Junonia limonias, common emigrant Catopsilia pomona, tawny coster, Acrea terpsichore, and blue tiger Tyriomala limnius. Definition. Migration in Lepidoptera means a regular, predictable movement of a population from one place to another, determined by the seasons. 2. There is no unambiguous definition of migratory butterfly or migratory moth, and this also applies to proposals to divide them into classes. 3. Migration means different things to behavioral scientists and ecologists. The former emphasize the act of moving, whereas the latter discriminate between whether the movement has been ecologically significant or not. Migration may be viewed as a behavioral process with ecological consequences. 2. Migration in Lepidoptera takes place in 2 of the 3 modes of migration identified by Johnson, 1969. In the first mode, also Johnson's first, the Lepidoptera move in one direction in their short lifespan and do not return. An example is the pierrot butterfly, Asha monist, which breeds in Florida but sometimes migrates along the coast up to 160 kilometers to breed in more suitable areas. 2. In the second mode, Johnson's third, migration takes place to a place of hibernation or estivation where they undergo diapause and the same generation survives to return. The classic example is that of the nymphalid monarch butterfly, Danuus plexippus. 2. Adventive. Species that are recorded in unexpected areas, adventive species, are not considered to be migratory species, because these did not leave their habitat on their own strength. Examples are species that are imported as egg or caterpillar alongside of their host plants or individuals that were reared by a collector but have escaped. An example of an introduced species is Galleria melonella, which is found all over the world, because it is reared as food for captive birds and reptiles. At times it is difficult to decide if a species is adventive or migratory. Migratory species like Chrysodeixis chalcites and Helicoverpa armigera would be able to reach Western Europe on their own, but are also common in greenhouses. Seasonal migration. Australian species Agrotis infusa spends the summer as an imago in the Australian Alps, and is sometimes found in buildings in large. Species that are recorded in unexpected areas, adventive species, are not considered to be migratory species, because these did not leave their habitat on their own strength. Examples are species that are imported as egg or caterpillar alongside of their host plants or individuals that were reared by a collector but have escaped. An example of an introduced species is Galleria melonella, which is found all over the world, because it is reared as food for captive birds and reptiles. At times it is difficult to decide if a species is adventive or migratory. Migratory species like Chrysodeixis chalcites and Helicoverpa armigera would be able to reach Western Europe on their own, but are also common in greenhouses. Seasonal migration. Australian species Agrotis infusa spends the summer as an imago in the Australian Alps, and is sometimes found in buildings in large numbers. Lepidoptera migration is often seasonal. With species of which all individuals migrate, the population moves between areas in the summer and winter season or the dry and wet season. For species of which only part of the population migrates, seasonal migration is hard to determine. They can maintain themselves in part of their habitat but also reach areas where they cannot establish a permanent population. With species of which all individuals migrate, Lepidoptera migration is often seasonal. With species of which all individuals migrate, the population moves between areas in the summer and winter season or the dry and wet season. For species of which only part of the population migrates, seasonal migration is hard to determine. They can maintain themselves in part of their habitat but also reach areas where they cannot establish a permanent population. They only live there in the season that is most favorable for the species. Some of the species have the habit of returning to their permanent residence at the end of the season. Difference with bird migration.